So yesterday we have learned with the interphase, G1 phase, S phase, G2 phase, and at last we have learned with the G0 phase. Now the next topic is division phase. So you know that division phase is also known as M phase. And requires so it takes at least only 1 hour, and there will be 4 hours of this cell cycle. So previously we have learned with this that interphase stage is the longest phase. Maximum 95% of the duration of the cell cycle can be taken by the interphase. So division phase or M phase, it lasts up to 1 hour mainly. It is the most dramatic period. Mitosis is also known as equational division and it occurs in haploid and diploid cell both. So mitosis occurs in both haploid as well as diploid cell. So here what happened, that site mitosis is actually continuous procedure. Fine, mitosis is continuous procedure. So for our understanding purpose, we divide the mitosis into 4 phases. Fine, as you know that prophase, metaphase, anaphase and telophase. So here in mitosis, first karyokinesis occurs. That is division of nucleus. That means metaphase, anaphase, and telophase occurs. And then it is followed by cytokinesis, means division of cytoplasm. Fine. So here mitosis is divided into two phases, karyokinesis, fine, and then cytokinesis. Now let us discuss in the one by one phase in detail. Fine. So first is prophase. So prophase is mainly, you have to remember that condensation of chromosome occurs. So due to condensation we can see the chromosomes very easily under the microscope, and here in the prophase you have to remember that nuclear membrane disappear, nucleolus disappear. As a result, in the nucleus the whole material it is mixed in the cytoplasm as well. Fine. So here we can write nuclear membrane and nucleus are disappear. Fine. In the plant cell, the centrioles are absent. So without centriole, the chromosomes are divided. So here ester fibers are not formed. So without the ester fibers, anestral mitosis occurs. I mean to say that ester fibers are not originated from the centriole. Whereas in the animal cell, the centrioles are present. So ester fibers are useful. Fine. So these ester fibers are useful for division of the chromosomes. So it is known as estral mitosis or amphiestral mitosis. Here the chromosomes are attached with the help of the centromere in the center of it, and then here you know that during the M phase, yesterday if you remember that during the S phase, centriole is divided. So now here the divided centriole is moved towards the polar region. So one centriole is present in the north pole and another centriole which is present in the south pole, and from the centriole ester fibers are formed. So at the end of the prophase, at the end of the prophase, Golgi apparatus, endoplasmic reticulum, nucleolus and nuclear envelope I told you here. Fine. These all are absent to see under the microscope. So this is about the prophase. Now metaphase. Okay. See, here in the metaphase stage, completely condensation occurs. So we see the morphology of the chromosomes easily and clearly under the microscope. That's why in the 8th chapter, Fine. If you have remembered that during the metaphase stage, only we see the chromosomes clearly under the microscope. So here we observe, we study the chromosome at metaphase stage. Now, one plant is there, Colchicum autumnale. So this is belonging to Liliaceae family, or the lily family we have learned in the 5th chapter. Find morphology of the plant in the root, stem, leaf, root, flower, seed and families. We have learned this. So Colchicum autumnale. From this plant we obtain the colchicine. So in any type of the cultural medium where cell division occurs, so in this cultural medium if you add the colchicine, so this colchicine arrests the cell division at the metaphase stage. So due to this colchicine we can easily arrest or prevent the cell division at the metaphase stage, and we see the chromosomes easily under the microscope. Along with that, this colchicine is carcinogenic also. Carcinogenic means cancer creating, and it is also mutagen, means it creates the mutation. So if our body is coming in contact with the colchicine, so might be genetic disorder occurs and mutation occurs, right? So that mutation, I hope you know that word, that the nitrogen base alteration, at the time of the cell division, the nitrogen base alteration, and it creates the change in the genetic makeup, and due to that the character which is shown by the same DNA, it can be altered. So this type of the condition is known as mutation. Okay, so colchicine is chemical mutant, mutagen. Fine. Chromosomes consist the kinetochores. So kinetochore are connected to bipolar spindle fibers and arranged in the center side. So arrangement of all the chromosomes in the center of the cell to form the metaphase plate or equatorial plate. So all the chromosomes are arranged in the center of the cell. Okay, so this is about the metaphase. Now anaphase. Okay, here what happened, as you know, that during the anaphase stage, division of the centromere occurs. So here, due to the division of the centromere, along with that, shortening in the length of the bipolar spindle fibers. So first of all, centromere is divided. So dear all students, please remember that centromere is divided in this anaphase. But where in the meiosis division, in anaphase 1, no centromere division, whereas in anaphase 2, centromere division occurs. Please remember that. Okay, again, in meiosis division, at anaphase 1, fine, centromere is not divided, whereas in the anaphase 2, centromere is divided. So here, due to the division of the centromere and shortening in the length of the bipolar spindle fibers, so half of the chromosomes are moved towards the polar region. So number of chromosomes become double in this cell. So here in the anaphase, the chromosomes become double. So up to right now, the chromosome numbers are same, but DNA material become double. The chromosomes are pulled towards the polar region, okay, due to shortening in the length of the bipolar spindle fibers. Now, the telophase. The last phase of the mitosis. Telophase is also known as reverse prophase. So the changes which are occur in the prophase, exactly opposite changes occur here. So here what happened? Nucleolus and nuclear envelope forms new Golgi apparatus and endoplasmic reticulums are formed. from nucleolus organization region. Fine. So from non-nucleolus organization region, nucleolus is synthesized here at the end of, see, this telophase is the ending of the karyokinesis. So at the end, two nucleus are formed which consist same number of chromosomes like parental cell. Okay, so here this is about the karyokinesis. So cytokinesis we will take it in the next lecture.